There are molecules circulating in your bloodstream that don't carry energy or nutrients, but rather information. Information that controls the functioning of different cells throughout your body. These molecules have directed your development from your mother's womb, through childhood, right up to today. And these molecules will continue to influence the functioning of your body for the rest of your life. Some of these molecules are released into the bloodstream in response to commands from the nervous system. Others in response to messages carried by other molecules. These molecules, in all their different shapes and sizes, are called hormones. Most hormones are produced and secreted into the bloodstream by small, specialized organs called glands, though a few are produced by major organs like the kidneys. The more than 50 hormones produced by the endocrine glands and various other organs of the body fall into one of 4 chemical classes: modified amino acids, peptides and proteins, steroids, and prostaglandins. The relatively few hormones synthesized through the modification of a single amino acid include epinephrine or adrenaline, norepinephrine or noradrenaline, and the thyroid hormone thyroxine. There are a large number of hormones made up of amino acid chains, ranging from a few to over 100 amino acids in length. These hormones are referred to as peptide hormones. Insulin, antidiuretic hormone, all the hormones produced by the hypothalamus and the pituitary glands, as well as many others, are considered peptide hormones. Steroid hormones, or steroids, all have a chemical structure like that of cholesterol, from which most are synthesized. Steroid hormones are secreted by the ovaries and placenta, estrogen and progesterone, the testes, testosterone, and the adrenal cortex, aldosterone. Nearly every type of cell in the body produces prostaglandins, which consist of 2 fatty acid carbon chains attached to a 5-carbon ring. Their biological effects include the lowering of blood pressure and the contraction of smooth muscle, such as that found throughout the gastrointestinal tract. Prostaglandins aren't considered true hormones, but are often referred to as local hormones, because they only act over a short distance before being broken down by enzymes in the blood. Though once in the bloodstream, hormones can potentially reach nearly every cell in the body, they only act on target cells that have a receptor for them. Cells lacking a receptor for a given hormone are unaffected by that hormone. Further, a given hormone can affect different target cells in our body differently. For example, the hormone epinephrine causes target cells in the liver to convert proteins and fats into glucose, while it causes target cells in the pancreas to release the hormone insulin. Depending on the hormone, a target cell's hormone receptors may be located either on the cell membrane or inside the cell, usually within the nucleus. Most peptide hormones, along with the modified amino acids epinephrine and norepinephrine, cannot cross the phospholipid bilayer of cell membranes. Instead, they must interact with protein receptors protruding from the cellular membrane of target cells. Generally, hormones that bind to surface receptors trigger rapid short-term responses. For example, some surface receptors are linked directly to protein channels that open immediately when a hormone binds to the receptor. A case in point is heart muscle cells, in which protein channels open instantly as epinephrine molecules bind their receptors, causing calcium ions to flow in and strengthen their contractions. In secondary messenger systems, the binding of a hormone to a receptor causes the receptor to change shape and trigger biochemical reactions that modify cell activity. One reaction often triggered is conversion of ATP to cyclic AMP, a nucleotide that regulates many cellular activities. Cyclic AMP thus acts as a second messenger, transferring the signal from the first messenger, the hormone, to molecules within the cell. The cyclic AMP initiates a series of reactions inside the cell. Each new reaction involving an increasing number of molecules amplifies the original signal. Results vary with the type of target cell. For example, channels may be opened in the cell membrane or substances synthesized or secreted. In smooth muscle cells, like those of the gastrointestinal tract, the protein calmodulin acts as a second messenger. The binding of particular hormones to receptors on the smooth muscle cells opens calcium channels that allow calcium ions to flow into the cells and bind to calmodulin molecules and change their shape. The altered calmodulin activates enzymes that in turn cause myosin and actin proteins to interact and initiate the contraction of smooth muscle cells that pushes materials through the GI tract. Steroid and thyroid hormones tend to initiate long-term responses that involve altering the activity of certain genes in a cell. Being soluble in lipids, steroid and thyroid hormones can diffuse through cell membranes and bind to receptors inside the cell. The resulting receptor hormone complexes bind to DNA and initiate the transcription of messenger RNA from specific genes. The messenger RNA then moves into the cytoplasm and directs the synthesis of new proteins, for example, enzymes involved in cell growth, metabolic activity, or in the case of cells in the testes, initiating the process of meiosis and the production of sperm cells during puberty. The release of hormones is usually regulated through negative feedback, in which the secretion of a hormone causes effects in target cells that inhibit further secretion of that hormone. Negative feedback is an important way of maintaining homeostasis, that is, keeping conditions within the body relatively constant over time. For example, it is important to maintain the level of glucose in your blood at about 100 milligrams per 100 milliliters. After a normal fast food meal with a large Coke, your blood glucose levels undoubtedly shoot up well beyond the normal level. Receptors on specific cells in your pancreas detect the increased blood glucose level, and as a result, these cells secrete the hormone insulin into your bloodstream. The insulin molecules, in turn, bind to target cells throughout the body and cause them to increase their intake of glucose. This results in glucose levels in your blood going back down to normal levels. Detecting this, the pancreatic cells that secrete insulin stop their release of insulin into the bloodstream to ensure that glucose doesn't drop below normal. Occasionally, positive feedback controls hormone release. Contractions of the uterus early in childbirth causes the release of oxytocin by the posterior pituitary. The oxytocin stimulates stronger contractions of the uterus, in turn causing more oxytocin release, and so on, creating a positive feedback cycle that continues until the infant is finally pushed out into the world. Mammals, including humans, have two types of glands: exocrine glands and endocrine glands. Exocrine glands produce secretions that are released outside the body or into the digestive tract through tubes or openings called ducts. The exocrine glands include the sweat and sebaceous, oil-producing glands of the skin, the lacrimal, tear-producing glands of the eye, the mammary, milk-producing glands, as well as glands producing digestive secretions. Endocrine glands release their hormones within the body and generally consist of clusters of hormone-producing cells embedded within a network of capillaries. These cell clusters secrete hormones which enter the surrounding capillaries via diffusion and are then distributed throughout the body by the bloodstream. Our understanding of hormonal control in our cells and other mammals is far from complete. Endocrinologists are continually discovering new hormones and new roles for known hormones. However, Science has been aware of the major endocrine glands and organs for many years. These include the hypothalamus-pituitary complex, the thyroid and parathyroid glands, the pancreas, the sex organs, and the adrenal glands. The hypothalamus, located at the base of the brain, contains clusters of neurosecretory cells that synthesize peptide hormones, store them, and then release them when stimulated. The pea-sized pituitary gland hangs from the hypothalamus and consists of two distinct lobes, the posterior pituitary and the anterior pituitary. The hypothalamus controls the release of hormones from both lobes. The anterior pituitary is a true endocrine gland, composed of several types of hormone-secreting cells enmeshed in a network of capillaries. The posterior pituitary, on the other hand, is an outgrowth of the hypothalamus, containing the endings of neurosecretory cells whose cell bodies are located in the hypothalamus. In the posterior pituitary, the cell endings of these neurosecretory cells intermesh with capillaries into which they release hormones into the bloodstream. The hypothalamus synthesizes two peptide hormones that are stored and released from the posterior pituitary: antidiuretic hormone, ADH, and oxytocin. Antidiuretic hormone works in conjunction with the central nervous system to prevent dehydration and maintain the proper balance of water in the blood. For example, if you ran several miles on a hot sunny day and lost a half liter of water through perspiration, nerve sensors in your hypothalamus would respond to the loss of water in your bloodstream by triggering the release of antidiuretic hormone, ADH, from your posterior pituitary gland, and issue a sensation of thirst that would direct your behavior. The ADH would travel to the kidneys and cause them to reabsorb more water and produce a very concentrated urine. The sensation of thirst would cause you to drink water shortly after finishing your run and replace the water lost through sweating. Once the sensors in the hypothalamus note that the water balance in the blood is restored, the secretion of ADH stops. Urine production returns to normal, and the sensation of thirst stops. This illustrates just one of several examples of endocrine glands interacting with the central nervous system to maintain the balance of vital materials in the body. As we saw earlier, oxytocin is involved in the contractions of the uterus during childbirth. After childbirth, the release of oxytocin is stimulated by the sound, the presence, or suckling of a newborn. This release of oxytocin triggers the contraction of muscle tissue within the breasts during breastfeeding and causes the ejection of milk from the sac-like milk glands into the nipples. 4 of the 6 major peptide hormones produced by the anterior pituitary help regulate the hormone production of other endocrine glands. 2 of these, follicle-stimulating hormone, FSH, and luteinizing hormone, LH, stimulate the production of sperm and testosterone in males, and eggs, estrogen, and progesterone in females. Thyroid-stimulating hormone, TSH, stimulates the release of hormones from the thyroid gland, while adrenocorticotropic hormone, or ACTH, causes the release of hormones from the adrenal cortex. Prolactin and growth hormone are the remaining 2 hormones produced by the anterior pituitary. In conjunction with other hormones, prolactin stimulates the development of the mammary glands during pregnancy. The suckling of a newborn further stimulates the release of prolactin and, in turn, the production of milk. When an infant stops suckling, the release of prolactin also stops, and, along with it, the production of milk. Growth hormone, which regulates body growth, acts on all the body's cells, increasing protein synthesis, fat utilization, and the storage of carbohydrates. As humans go from infancy to adulthood, growth hormone stimulates bone growth. Much of the variation in height in adult humans is due to differences in the secretion of growth hormone during childhood and adolescence. Too little growth hormone causes some forms of dwarfism, whereas too much can cause gigantism. Dwarfism can be treated by administering growth hormone to children with underactive pituitary glands. In adulthood, growth hormone continues to be secreted and plays an important role in regulating protein, fat, and sugar metabolism in the cells of the body. Neurosecretory cells within the hypothalamus produce at least 9 peptide hormones that regulate the release of hormones from the anterior pituitary. Releasing hormones stimulate, while inhibiting hormones suppress the release of a particular pituitary hormone. Releasing and inhibiting hormones secreted in the lower portion of the hypothalamus travel the short distance down the pituitary stalk to the endocrine cells of the anterior pituitary. Because releasing and inhibiting hormones are secreted very close to the anterior pituitary, they are produced in minute quantities and are extremely difficult to isolate and study. American endocrinologists William Shalley and Roger Guillemin, who shared the Nobel Prize in 1977 for describing several of these hormones, had to obtain the hypothalami of hundreds of thousands of sheep and pig brains from slaughterhouses in order to extract sufficient hormones for analysis. The thyroid gland is located at the base of the neck, in front of the trachea or windpipe. Embedded in the back of the thyroid are the 4 small disc-shaped parathyroid glands. The thyroid produces the hormones thyroxine, triiodothyronine, and calcitonin. Both thyroxine and triiodothyronine are modified amino acids that contain iodine and raise the metabolic rate of most body cells. In juvenile animals, including humans, thyroxine and triiodothyronine help regulate growth, stimulating both metabolic rate and development of the nervous system. The generalized elevation of metabolic rate caused by the release of thyroxine increases oxygen consumption and heart rate and stimulates the synthesis of enzymes that break down glucose and provide energy. In adults, an elevated metabolic rate seems to be involved in regulating body temperature and responding to stressful situations. For example, exposure to cold increases the production of thyroid hormone, resulting in an elevated metabolic rate that helps maintain body temperature. Levels of thyroxine in the bloodstream are finely tuned by negative feedback loops. Thyroxine release is stimulated by thyroid-stimulating hormone, TSH, from the anterior pituitary, which in turn is stimulated by a releasing hormone from the hypothalamus. The release of TSH from the pituitary is regulated by thyroxine levels in the blood. High concentrations of thyroxine inhibit the secretion of both releasing hormone and TSH, thus inhibiting further release of thyroxine by the thyroid. A number of diseases may result if thyroxine levels get out of balance. In hyperthyroidism, high body temperature, profuse sweating, weight loss, irritability, and high blood pressure result from excess metabolic activity due to high levels of thyroxine in the blood. On the other hand, symptoms of hypothyroidism include weight gain, lethargy, and intolerance to cold, resulting from lowered metabolic activity owing to reduced levels of thyroxine in the blood. Iodine is critical to the synthesis of thyroxine. As a result, an iodine deficiency leads to a reduction in thyroxine levels that causes the thyroid to grow dramatically in an effort to restore normal thyroxine levels. The enlarged thyroid bulges from the neck, producing a condition called goiter. Once common, goiter has all but been eliminated in developed countries by the widespread use of iodized salt, but remains common in some undeveloped areas, affecting as much as 5% of the population worldwide. Because of reduced levels of thyroxine in the blood, victims of goiter have symptoms very much like individuals who suffer from hypothyroidism. Calcitonin, a peptide protein produced by the thyroid, acts in conjunction with parathyroid hormone, or PTH, secreted by the parathyroids to control the concentration of calcium in the blood. Calcium is essential for nerve, muscle, and other functions, so bodily calcium concentrations must be kept within narrow limits. If blood calcium levels become too high, the thyroid releases calcitonin to inhibit the release of calcium from bones. In response to low blood calcium, the parathyroids release parathormone, which causes bones to release calcium. In pregnant and lactating women, the parathyroids increase in size, and parathormone output goes up to meet the demand for extra calcium imposed by a developing fetus and later milk production. The pancreas consists largely of exocrine tissue that produces digestive juices released into the small intestine via the pancreatic duct. However, endocrine cells are scattered throughout the pancreas in about 2 million cell clusters called pancreatic islets, or islets of Langerhans. Each pancreatic islet has islet cells that produce the peptide hormone insulin, while other islet cells produce glucagones, another peptide hormone. Insulin and glucagon work in opposition to regulate the concentration of glucose in the blood. Like calcium levels, blood glucose levels are also critical, as glucose is a major fuel for cellular respiration and an important source of carbon skeletons for the synthesis of other organic molecules. When blood glucose rises, for example after a meal, insulin is released. Insulin causes most of the cells of the body to take up glucose and either metabolize it for energy or convert it to fat or glycogen, most of which is stored in the liver and skeletal muscles. Insulin further decreases blood glucose levels by inhibiting the conversion of fats and amino acids to sugar, and by slowing down the breakdown of glycogen into glucose in the liver. When blood glucose levels drop, for example after skipping breakfast or a cross-country run, glucagon is released and signals liver cells to break glycogen, amino acids, and fatty acids down into glucose, and to start slowly releasing the glucose into the bloodstream. Deficiencies in insulin production or loss of response to insulin by target cells results in diabetes mellitus, a condition in which blood glucose levels are high and fluctuate wildly with sugar intake. In an effort to compensate, the kidneys begin to expel glucose and with it excessive amounts of water, leading to frequent urination and the persistent thirst of diabetics. The functional lack of insulin in diabetics causes the body to rely much more heavily on fats as an energy source, leading to high circulating levels of lipids, including cholesterol. Severe diabetes causes fat deposits in the blood vessels, resulting in high blood pressure and heart disease. Fatty deposits in the small vessels of the retina can lead to blindness, while fatty deposits in the small vessels of the kidneys can lead to kidney failure. The gonads, testes in males and ovaries in females, are important endocrine organs, producing steroid hormones that affect growth and development, as well as regulating reproductive cycles and behaviors. There are 3 major categories of gonadal steroids: androgens, estrogens, and progestins. Both males and females have all 3 types, but in different proportions. The testes primarily secrete androgens, the most important being testosterone. Androgens are generally involved in the development and maintenance of the male reproductive system. Early in embryonic development, the presence or absence of androgens determine the sex of an individual. If present, a male develops. In its absence, a girl. Later, during puberty, high concentrations of androgens initiate the development of human males' secondary sexual characteristics, such as facial hair, increased musculature, and deeper voice. The ovaries primarily secrete estrogens and progesterone. Like androgens in males, estrogens and progesterone in females are largely involved in the development and maintenance of the female reproductive system. As increased levels of estrogens and progesterone begin to circulate in females during puberty, they cause the development of secondary sexual characteristics, such as enlarged breasts, broadening of the hips, increased subcutaneous fat, development of the uterus, and initiation of the menstrual cycle. On top of each of the body's 2 kidneys lies an adrenal gland. Structurally and functionally, each adrenal gland actually consists of a gland within a gland. On the inside lies the adrenal medulla, and on the outside the adrenal cortex. The adrenal medulla consists of secretory cells derived during development from nervous tissue, and its hormone secretions are controlled directly by the sympathetic nervous system, which is charged with preparing the body for emergencies. Appropriately, The adrenal medulla produces two modified amino acid hormones, epinephrine and norepinephrine, also called adrenaline and noradrenaline, in response to fear, extreme pleasure, and stress. These hormones prepare the body for emergency action by increasing heart and respiratory rates along with blood glucose levels while directing blood flow away from the digestive tract and toward the brain and muscles. The adrenal cortex forms the outer layer of the adrenal glands. The cortex secretes three types of steroid hormones called glucocorticoids that promote the synthesis of glucose from fats and proteins, raise blood pressure, and reduce inflammation. Glucocorticoid release is stimulated by ACTH from the anterior pituitary. ACTH release, in turn, is stimulated by releasing hormones produced by the hypothalamus in response to stressful stimuli including trauma, infection, or exposure to extremes of temperature. In some respects, glucocorticoids act similarly to glucagon, raising blood glucose levels by stimulating glucose production and promoting the use of fats instead of glucose for energy. The adrenal cortex also secretes aldosterone, which regulates the sodium and water content of the blood. Sodium ions derived from dietary salt are the major positive ions in blood and extracellular fluid. The movement of sodium ions across cell membranes is critical in many cellular events, including the generating of electrical signals by nerve cells. If blood sodium falls, the adrenal cortex releases aldosterone, causing the kidneys and sweat glands to retain sodium. This increase in sodium also increases blood volume, as water is drawn from the body's cells by osmosis. Thus, aldosterone is also released by the adrenal cortex in response to a loss of blood due to an injury. Finally, the adrenal cortex produces sex hormones, mainly androgens similar to testosterone, and small amounts of the female hormones estrogen and progesterone. It has been proposed that the androgens produced by the kidneys are what produce the sex drive in adult females. It is known that tumors of the adrenal medulla sometimes lead to excessive androgen release, causing masculinization in women. Many of the bearded ladies who once appeared in circus sideshows probably suffered from this condition. The pineal gland, located between the brain's two hemispheres, just above and behind the brainstem, resembles a pea-sized pine cone. While many of its functions are poorly understood, it is known that the pineal secretes the hormone melatonin in a daily rhythm regulated in mammals by light sensors in the eyes. In other vertebrates, such as frogs and the ancient tuatara lizard, the pineal itself contains photoreceptive cells, and the skull above it is thin, so the pineal can detect sunlight and thus day length directly. By responding to day lengths characteristic of different seasons, the pineal appears to regulate the seasonal reproductive cycles of many mammals. Despite years of research, the function of the human pineal gland and melatonin is still unclear. One hypothesis is that melatonin, secreted by the pineal gland, influences sleep-wake cycles, and that improper pineal function may contribute to a type of depression called seasonal affective disorder, or SAD. Seasonal affective disorder usually occurs during the short days of winter. Interestingly, these symptoms of depression can often be dramatically reversed by exposing patients to high-intensity, broad-spectrum light for short periods of time each day during the winter months. Such exposure apparently works by reducing the amount of melatonin released into the bloodstream by the pineal gland. The thymus, located in the chest cavity behind the breastbone, besides producing white blood cells, produces the hormone thymosin. Thymosin stimulates the development of specialized white blood cells, called T cells, that play an important role in the immune system. The thymus is extremely large in infants, but decreases in size after puberty as the immune system becomes well established. The kidneys, as we've seen, play a central role in maintaining sodium balance and blood volume, as well as carrying out its major function of filtering the blood. Recently, it has been recognized that the kidneys act as important endocrine organs as well. Drops in the oxygen content of the blood cause the kidneys to produce the hormone erythropoietin, which increases red blood cell production. The kidneys also produce a second hormone, renin, in response to low blood pressure, such as may be caused by bleeding. Renin is often referred to as a hormone, but is actually an enzyme that catalyzes a reaction that produces the hormone angiotensin from proteins in the blood. Angiotensin raises blood pressure by constricting arterioles. It also stimulates aldosterone released by the adrenal cortex, which, as we've seen, causes the kidneys to return sodium to the blood, which in turn increases blood volume. The heart seems an unlikely endocrine organ. But in 1981, a substance extracted from atrial tissue in the heart was found to cause the kidneys to expel more sodium and water in the urine when injected into rats. 2 years later, the active substance atrial natriuretic peptide was described and its amino acid sequence determined. ANP is released by cells in the atria of the heart when blood volume increases to the point where it causes excessive and potentially damaging distension to the heart. Atrial natriuretic peptide causes reduction of blood volume by decreasing the release of both ADH and aldosterone. 5 years after it was first detected, clinical trials of synthetic ANP were started to test its value in treating high blood pressure and related problems.